Welcome back, students. This is Module 2.1.3, Cellular Structure. The first structure we're going to consider is the cell membrane, also known as the plasma membrane. We'll preferentially use the first term. The cell membrane defines and protects the cell. It controls transport in and out of cells. Of course, it is made of phospholipids. We call it a phospholipid bilayer because of hydrophobic forces when in a watery environment. This will form a bilayer naturally. And there are also proteins stuck in there. We'll show you a diagram in just a moment to kind of drive home this structure a little bit more. Cell membranes are found in all cells, both prokaryotic and eukaryotic. Recall, our prokaryotic cells, are they bacteria, or are eukaryotic cells bacteria? If you said prokaryotic, you would be correct. So those are bacteria. But both prokaryotic and eukaryotic cells have cell membranes. And you should recall the relationship between the cell wall and the cell membrane. If not all cells have a cell wall, but if they do, it is found outside the cell membrane. The cell membrane is present beneath the cell wall. And here is a diagram of a cell membrane. You remember the phospholipid bilayer. You can see these little round structures on both sides and then the little tails there. The round structures are the phosphate heads, and they are polar or hydrophilic, water-loving, and the tails are hydrophobic. These are fatty acid tails. And so in a watery environment, there'd be water out here and water inside here, and the tails are hydrophobic, and so they are avoiding the water, and they turn inward and naturally form a bilayer. And this would be in the form of a sphere. So you can see that they would avoid the water in that way. And in the cell membrane, there are, in addition to just phospholipids, there are other structures, these large blobs here, this purple one and this blue one. These are proteins. And then we can see that the little yellow structures here, this should look somewhat familiar to you, perhaps, if you remember steroids. This is cholesterol, and cholesterol helps to maintain proper membrane fluidity. So this just gives you a little bit of an introduction to the cell membrane. We will come back and look at it in even more detail in a future unit. Other structures that are found inside the cell include cytoplasm, the semi-fluid matrix that fills the interior of the cell. This is the site of many chemical reactions and many cellular structures. And the cytoplasm and the cell membrane we spoke of previously are present in all cells. The cell wall, however, a cell wall, however, is not present in all cells. It is a protective layer or supportive layer outside the cell membrane, present in some cells as animals and some protists lack them. This picture on the lower right is showing the surface view of a plant cell wall, and you can actually see the fibers of cellulose that are making this up. This little drawing here is meant to show two, for example, plant cells that are abut. The yellow line here is the cell membrane, and the thick layer outside it is the cell wall. And this is the way two cells come together, the cell walls and the cell membranes inside of that. Now we're going to look at some other structures. Another structure that's found in both prokaryotic and eukaryotic cells is a structure called the ribosome. Ribosomes are small particles that are found in the cytoplasm, and they are the site of protein synthesis. So this is where proteins are made, where amino acids are put together to make the long polymer proteins. And ribosomes are present in all cells. A structure which is found in some cells and not in others is a flagellum. A flagellum, many students want to think of it as a tail that pushes a cell, a single cell. For example, the tail of a spermatozoan or a sperm cell. This is how it works. But there are some other flagella which actually kind of pull. And the flagellum is simply a protein extension that's used for movement, found in some cells and not in others. Something that we do find in all cells is a cytoskeleton. This is a network of protein fibers in the interior of the cell. Many of you might be thinking of a cell as a squishy structure, kind of like a big water balloon or something like this. And that just is not the case. Cells are more organized, and they have this network of protein filaments inside that help to maintain the shape of the cell and anchor organelles if they're present, as in eukaryotes. And these protein filaments also serve almost as rails for transport. Things are moved around the cell in a controlled way by moving on the filaments of the cytoskeleton. So it's actually a very interesting kind of arrangement. We want to discuss organelles, and I told you we would revisit the definition. Here it is. An organelle is a subcellular membrane-bound structure with a specific function. Subcellular means it's smaller than a cell. Membrane-bound means it's surrounded by a membrane, and then it has a definitive function. And organelles actually provide compartments in eukaryotic cells, and you can have specialization and separation with organelles that's not possible in their absence. Remember that eukaryotes have them and prokaryotes lack organelles. And so prokaryotes have to maintain many functions, but what they do is they perform their cellular functions either in the cytoplasm or on the interior of the cell membrane. They don't have a lot of other membranes like prokaryotes do. The organelles that we're going to discuss now are found only in eukaryotes and not in prokaryotes. So here we go, organelles. The first organelle we're going to discuss is a nucleus. Recall that the nucleus is a spherical structure that contains DNA. It's surrounded by a double membrane called the nuclear envelope or the nuclear membrane. And of course, because the nucleus contains DNA, DNA controls both the short-term and long-term functions of the cell. The long-term functions would be mostly heredity. The short-term are both mostly biochemical, metabolic functions, making proteins at the proper time and in the proper amounts. So if you look at this, this is a scanning electron micrograph, and this is zoomed in on the nucleus. So this highlighted region is the nucleus. Out here it would be the cytoplasm with organelles and different things happening. Associated with the nucleus are often these little spots or dots. And in this case, what we call such a spot or a dot a nucleolus. And a nucleolus is a concentration of a certain type of RNA that makes ribosomes. And so if we look on this particular photomicrograph, there is the nucleolus highlighted. And you may have multiple nucleoli in the same cell. And so that can be kind of confusing, the similarity of nucleus and nucleolus, but nucleolus is like a diminutive. It means like a small little structure inside the nucleus. Another organelle is endoplasmic reticulum, a big old name. Actually, I like this name because it's very descriptive, and if you break it down using etymology, it makes a lot of sense. ER for short, endoplasmic reticulum. Endoplasmic means inside, endo means inside, and plasmic refers to cytoplasm, and reticulum means network. And so it's very interesting. Does anyone know the biggest snake in the world, the longest snake in the world? It's the reticulated python, and it has this network kind of pattern on it, and so that's the association with reticulum, reticulated. And so what endoplasmic reticulum is, as an organelle inside eukaryotic cells, it is a network of flattened membranes that provides transport and functions in lipid synthesis and protein synthesis. Now, this is a very kind of confusing thing, so I've tried to make it a little clear. Let's see how we do. There are two types of endoplasmic reticulum: smooth, that lacks ribosomes, and rough, that has ribosomes. So we have smooth ER and rough ER. And so let's look at a picture and see if we can further understand this. You're going to have to concentrate and review this a little bit. But so there we can see that this is in the diagram at the bottom, this is smooth endoplasmic reticulum. And so that's one of the two types. And it has two functions. It functions in transport, because both types of ER do that. And smooth endoplasmic reticulum also functions in lipid synthesis. Now, if we look at the other type, this is rough endoplasmic reticulum. Look at the diagram at the bottom. See, it is the rough. And again, it has two functions. One of the functions of rough ER, rough endoplasmic reticulum, is transport, and the other is protein synthesis. Of course, that makes sense if you think about it. What makes rough endoplasmic reticulum rough is the presence of ribosomes on the outside, these little dots here. And those little dots are ribosomes, and that's the site of protein synthesis. So that makes a lot of sense. And endoplasmic reticulum really has a form which lends itself to transport. It's this tubular structure, so proteins can be made and transported inside, lipids can be made and transported inside. And you can see that this purple structure here is the nucleus, so the endoplasmic reticulum is often closely associated with the nucleus. And the more proteins a cell needs, the more endoplasmic reticulum it will have, especially rough endoplasmic reticulum. Let's look at another organelle, and this is the Golgi apparatus, or the Golgi body. And I told you I like the name endoplasmic reticulum. As much as I like that name, I dislike this name, because this is named after some Italian microscopist who discovered the Golgi apparatus, and now we have to call this after him. It doesn't make any sense. It's not descriptive, so I don't like descriptive names. The Golgi apparatus, however, is an organelle found, again, only in eukaryotic cells, and it consists of stacks of flattened membranes that package and label cell products, especially proteins. Sometimes we can think of the Golgi apparatus as the mailroom, because it kind of puts the address label on. And so it's putting these labels onto especially proteins. Now here's a diagram. Let's take a moment and see if we can understand this. This is this kind of orangey structure to your left is the endoplasmic reticulum, and this is rough ER. You can see because it has these little ribosomes on it, so it makes protein and transports things, so things can move through it. And when they get to the end of it, it forms little vesicles, and these are transport or transition vesicles, and they will carry the protein, and it will come over and fuse with this structure here in the center of the illustration. This is the Golgi body, and so it fuses, and it can move through, and then it will pinch off on the other side after a proper label has been put on that protein, and then it will go and fuse with the cell membrane on the outside of the cell, and then it will release the proteins with the labels, and the labels will allow the proteins to go to the proper cell in the body where they need to go. So the Golgi performs an important function. An additional organelle is the lysosome. And we can use an etymological approach here. Lys means to split, and some means body. So these are bodies which can cause splitting. This is because they are membranes that are filled with digestive enzymes, and they help to digest worn-out organelles and foreign bodies. And so lysosomes do contain these digestive enzymes. Another organelle found in eukaryotic cells is the mitochondrion. I'm sorry, I didn't show you the picture of the lysosome. Oh, sure, it's very informative. Another organelle is the mitochondrion, and it's an oval structure in which the reactions of cellular respiration occur. And what these reactions do is they take food that the cell has inside it and releases the energy inside it. It transforms it into ATP, which is the energy source. In high school, you might have learned that the mitochondrion is sometimes called there's an illustration of the mitochondria. It's sometimes called the powerhouse of the cell. And the origin of the mitochondrion is kind of interesting. It is believed to have originated when one cell engulfed another, and the engulfed cell continued to live inside the larger cell, and the mitochondria are the engulfed cells. This is called endosymbiosis, and so we'll talk more about this in just a moment. So mitochondria are believed to have originated from bacteria by endosymbiosis. Mitochondrion is singular, mitochondria is plural. Another organelle that's found inside eukaryotic cells and this is found only in some eukaryotic cells, are chloroplasts. And chloroplasts are organelles that contain the green pigment chlorophyll, and these are able to undergo photosynthesis, and that transforms light energy into chemical energy, into chemical bonds in food. And so they're not producing energy, but they are transforming it from light to chemical bonds. And and here's what the chloroplast looks like. When we study photosynthesis, we'll learn some more of the structures here. But for now, it's just these green spheres is all we're going to learn. But like mitochondria, chloroplasts are believed to have originated by endosymbiosis. So let's talk more about endosymbiosis, believed to be the origin of two organelles. This is showing time 1 and time 2. This little blue arrow separates them. So up here, this shows an aerobic heterotrophic prokaryote. In other words, a little bacterium, and it is engulfed by a larger cell, and time 2 it takes up residence and becomes a mitochondrion. Now, a separate event, this didn't happen in the same cell at the same time as is shown in this illustration. That's just for convenience, but it's showing a photosynthetic prokaryote, so a little bacterial photosynthesizing cell that was engulfed, and it continued to live inside the larger cell and became the chloroplast. And so this is a hypothesis, and there's evidence. Some people might even call it a theory, but the hypothesis of endosymbiosis explains the origin of mitochondria in chloroplasts. Now let's look at some of the evidences in support of this concept. Here shows the mitochondria and then shows the chloroplast. So let's look at some evidence. It includes that mitochondria and chloroplasts have their own DNA. This is very unusual and not typical of organelles. Other organelles all are coded for inside the nuclear DNA of eukaryotic cells. But to make more mitochondria, the mitochondria has to divide in two. It has its own DNA. The same with chloroplasts. So this is consistent with being a separate cell that was engulfed by a larger cell. Another piece of evidence is that both chloroplasts and mitochondria have a double membrane. They had their own cell membrane, and then when they were engulfed, a vacuole formed around, and that made a second membrane. And that's consistent with the idea of endosymbiosis. Another piece of evidence is the fact that endosymbiosis occurs today. We can have cells engulf other cells, and they can live inside for a very long time. So it's not like this is some impossible feat that we're hypothesizing. It's something that happens today. There's very strong evidence for it. Here, in fact, is showing a large protist with a bunch of green algae. These little spheres inside are all green algae. Hundreds of this green algae called chlorella that are inside this cell. So it's kind of interesting that this does occur today. And so that is the hypothesized origin of mitochondria and chloroplasts. That also ends our discussion of cell structures. So thank you, and we'll be back.